My brain hurts already.
Hello, everybody. Welcome to Q&A with A&V. I'm
And you
Vincent
tell me it's because
Racaniello.
I'm sulking, but it's not. It's because my brain hurts already.
Joining me tonight in true form from New York, Amy Rosenfeld. Welcome, Amy.
Hi, Vincent. How was your day today? Did you have a good day?
It was a day of viruses.
Okay.
Day of viruses. And yours was
Plus the
also
eye
a day
doctor.
of viruses. I saw
Plus
an eye
the
doctor.
mine is the eye doctor.
I taught a course, and then I did work, and then I'm here with you. Good way to round out a virus day.
Yes, but my brain hurts now.
Yes, the brain is hurting. Every time we have a new variant, our brains hurt. But let's start out with.
Where's your Q&A? You wanted your show and tell.
Yeah, I want to show these data as we start out here. So let's share the screen. This is from Hong Kong University. It is a press release from them, and it says a team of researchers has looked at infection of lung tissues removed for treatment of the lung, lower and upper respiratory tract, and they infected them with SARS-CoV-2 original virus, the Delta variant, and Omicron. And at 24 and 48 hours after infection, they take bronchus and lung specimens. All right, so bronchus a little higher up, lungs a little lower down, right? And they infect them, measure actually infectious virus. And I wish they had a zero time point, Amy, don't you?
You always need a zero time point.
Because you don't know what's going on between 0 and 24. Nevertheless,
well, you don't know how much stuck.
Yep.
How much stuck. There's
Omicron,
an absorption consequence.
So
right?
Omicron is at 10 to the 4 TCID 50s per mil, about 2 logs higher than the original in Delta in the bronchus. But then in the lung, it doesn't do as well, interestingly, the original virus.
Yeah,
so,
but
go ahead.
Yeah. Go ahead.
It just doesn't do as well in the lower tract, the lung tissues, compared to upper, the bronchus, right? That's the observation. So,
well, no, so I wouldn't say that.
Mm-hmm. Go tell me what you would say.
So I wouldn't say that, because if you look at the difference, the rate of increase between, since we don't have a time zero, you can't say anything. You can only draw the line starting at 24 hours, right? So it's the blue, right? So at 24 hours, it's 10 to the 4th. And then at 48 hours wait, you move the graph. Go back down.
Sorry.
So you move, at 48 hours, it's not even a full log above. Let's say that this 24-hour time point was the inoculum, since they cheated without time zero.
Yeah.
So it doesn't even go up a full log. But in the lung, if you said that the 24-hour time period was actually the time zero if they hadn't cheated, so it's at 10 to the 2, and now it's actually it actually is a full log, because it went over 10 to the 3. So their conclusion is erroneous.
So also, Amy, I'm wondering, and let me get us back here. I don't know about the quantification of this, right? This is per mil, but what is a mil? Is it equivalent in number of cells per culture? I would like to know per cell, wouldn't you? TCID 50 per cell, not per mil.
I don't know. We do PFU per mil.
Yeah, but usually you have the same number of cells. Anyway.
Well, you usually say that you've infected your monolayer at an MOI of 3, right? That's in the material method.
And you have equivalent numbers of cells, although here these are explants, so I don't know about equivalent numbers of cells. How would they do that, right?
Right, but I'm just saying that if you draw the line is not supporting their hypothesis or their conclusion.
I understand. So this is in review. We'll see if it comes out, actually, because it's...
Well, it should not come out without a time zero, because otherwise you can't say anything. Because I could argue that time point at 24 hours is all absorption, right?
Yeah.
Stickiness from absorption. And I don't like stickiness from absorption, so this is not good. But I doubt our colleagues will be vigorous in sending it back to the author, stating that they need to have a time zero to make any conclusion. Because right there, and say, take an algebra class, learn y equals mx It's all y equals mx plus b. Take an algebra class. Learn y equals mx plus b, because then you'll find that the slope of the line does not support your conclusion.
Are you banging your desk?
Sorry.
Because it comes through.
It was the cap to my soda spray, my soda stream bottle.
Okay. All right. The other bit of news. Do you want to tell them the other bit of news? What did we do yesterday, Amy?
Go to 4 mouse houses and be booted out of them all at the same time.
No, that's not what I was thinking about.
Okay, well,
how's your arm feeling? How's your arm feeling, Amy?
Not so good.
Is it sore?
It is very sore, very heavy, and now I have the chills.
You have chills. So what did you have put in that arm?
I was harpooned yesterday.
All right. So what Amy doesn't want to say is we both went for 3rd doses of vaccine yesterday. Based on the data out of Ontario last week that we discussed, where that 3rd dose really makes up for the inappropriately spaced 1st and 2nd doses. And I think it's not the whole story because it's antibody levels, and I'm very wary of that, but it's something. And we're not going to have disease for a while, so we thought it would be a good idea. So Amy originally got 2 Pfizers, and now she got a 3rd Pfizer. All they had at Columbia was Pfizer. So I got Pfizer, even though my first 2 were Moderna. So it doesn't matter. People who are asking, it doesn't matter. Just get everything you can.
The other thing is where did I see it? I sent it to you and Daniel, a quote that talks about it's all about the T cells. I sent it. Yeah. I sent the paper. What paper did I send? I sent you and Daniel.
It was an Atlantic article, wasn't it?
No, I thought it was an actual paper.
But we've been saying that for months, haven't we, Amy? It's all about the T cells ever since Sete came.
I don't know because I get told I'm wrong, and then I wait a couple of months and I forget, and then it all sounds very familiar, and then I don't know. It's just a big mess.
Amy, someone said that's why you have a headache, because of your booster. Exactly right.
No, I have a headache because I'm stressed out and there's a lot of stupidity in the world.
Anyway, folks, so I told you when we had the data, we would decide. And so we see the data. And I don't think it was the right thing 6 months ago, but I think it is now. So you make your decisions. I think listen to the last week's TWIV. It's good data out of Canada. It's a published peer-reviewed paper. All right, let's get to some questions here. Are there any steps besides vaccination and responsible distancing mask use that can help prevent or lessen the danger of long COVID? Any thoughts, Amy?
Since I don't understand how long COVID occurs, probably not. I mean, I don't know. Maybe you're supposed to do the traditional Jewish thing, get some chicken soup and rest. I don't know.
We don't know anything about long COVID. Yeah, that's the problem. So we would have no idea how to prevent it. There is a low level of long COVID after an infection of a vaccinated person. Daniel brought up that paper last week. Yeah, very low level in a study, but very low. So, I mean, getting vaccinated is important, but why it happens anyway, we don't know. We're not going to know for a long time, I bet. For sure.
I'm thinking it's going to be very hard to model in an animal.
Sure. Just like MECFS is very hard to model, right?
I didn't know that there was an animal model.
There isn't any for MECSF, no.
Yeah, I didn't think so.
Speaking of your favorite people, your favorite Peter Hotez claims today on CNN that these might be 5-dose vaccine primary series. We need to stop all and every infection. That's just ridiculous. It's just I think the 3 is enough, in my view. Amy, I thought you were going to shut him down on Twitter.
I don't shut anybody down. I'm not the Twitter police. There is a Twitter police.
Really?
There is this Twitter police. They took away Donald Trump's Twitter account and various other people.
That's
you
true.
Get a timeout. The lady, the QAnon lady at Georgia, she's gotten multiple timeouts. I don't know why the Twitter people haven't given Peter Hotez a 6-month to a year timeout. I'm not in charge.
Well, no, who's going to evaluate the science? I mean, the other stuff was politics, but who's going to evaluate Hotez's science? He's head of a vaccine institute. They're going to figure he knows what he's talking about, right?
Everybody makes mistakes.
Another question from Mark. The new preprint about major vaccines and antibody escape. How much generally does antibody protection mean as opposed to T and B cells? Well, that's the question.
No, he doesn't say opposed to the...
Well, that is supposed to be T. That is supposed to be T. T and B cell protection. So T cell, that's the key.
B cells are the antibodies.
Yeah. B cells are the antibodies. The T cells, in our view, are always protecting you because none of the variants substantially change in the sequences in the protein in the spike that are recognized by T cells. And that's why the prevention of hospitalization and death remains in the 90s with mRNA vaccines against even Delta. And I suspect it will be the same for Omicron.
Yeah, I just saw a tweet or something or other by like Cromer or somebody else who also is now being converted to seeing the light to the T cells.
I have someone's email that I have to respond to who said it was bogus, but we won't mention that here.
I already did. If it's the person that I'm thinking about.
So all you folks are really, people in the chat are talking about all these sorts of people, Kizmikia, Paul
sort of
Offit, Kizmikia, Paul Offit, Shane Karate, Sate. They have all been on Twiv, folks. All right, we have a minimal impact, or not, I shouldn't say impact. We have a minimal audience, nothing like the major outlets. Yet we really have the people, and they learn from us who to take, and we have good episodes with them. So we should have more people watching. So Friday is Paul Offit. In January will be Alessandro Sette. Who else do you want? Let us know.
Well, they have to be pre-approved by me, so it doesn't matter.
Are people totally overreacting to Omicron? If you're vaccinated, it seems like not such a big deal, especially 3 doses. Yes, I think it's overreaction, but it's driven by scientists, right? It's driven by scientists. Like, well, I'm not going to name names.
Thank God, yes. Thank God you've come and seen the light.
Yeah, the scientists speak to the journalists who just pass on their words.
Exactly.
Now, CNN talked to me at the beginning of the outbreak, and I wasn't hysterical enough, so they never had me back. Remember that, Amy?
I do remember. I do remember because our collaborator who no longer responds to my emails wrote us an email saying Sanjan Gupta or whatever his name is should beware. Vincent Racaniello is on the scene. Remember that?
I do.
Yeah, I don't know. It's fine. The more important thing is to tap. People should more tap into Twit, like however they want to do it. Get the word spread.
Hello, Frank. Welcome. Thanks for moderating. I didn't catch Frank earlier. Look at this. My work announced we have to show proof of booster today. New York City. Wow.
Not surprising.
Not surprising at all.
No. Although I wonder how long that's going to last as the mayor changes at midnight on January 1st.
You think it might?
Yeah, I don't think Eric Adams is. I think he's more mainstream than de Blasio, and probably will have a sounder policy.
Does he have to follow? Does he have to follow New York State policy?
Yeah, but like New York State doesn't really. Their policy usually goes hand in hand, but like I think. So there's a new police commissioner. It's the first woman, African-American woman, police commissioner of New York City. And I'm not sure that she is not fully, I'm not sure that Adams did not have a role in her selection. I'm not sure when her tenure begins, but that's kind of cool.
So I see later in the chat, someone says Joe Rogan had Peter McCullough on in a super spreader of misinformation. So Rogan is too chicken to have me on. I think he'd be totally chicken to have Amy on, or both of us. That would be something. But
I wouldn't go without you. Why would I go without you?
he is afraid to get the truth. I dare him to have us on. All right, Mike
Is
says
this like Double Dare? Can we have a
I'm
Double
just doing a
Dare?
regular dare, no double dare. That's
I don't even know what it means. But first of all, who is Peter McCollum?
I don't really know. I'm not
Okay.
familiar with
I'm not familiar with all these.
misinformation people.
Yeah.
Just the ones who used to be good, full of good information, who turned. They went to the dark side. All right. Outside of vaccines, what other reasons are there for the declining case fatality ratio? Vaccination. And
Also, there's some drugs and monoclonal therapy and stuff.
and also learning how to treat patients, right?
Yeah,
As
exactly.
Daniel says, what to do at different stages. Don't give steroids early, right? Don't give antivirals late. So yes, I think hospital care has a lot to do with it. Oh, so this is a question we got also. Are children 5 to 11 who got 2 doses of Pfizer protected? So they got the doses 3 to 4 weeks apart. So I think they need a third dose, don't you, Amy?
Yeah, I think so. Well, I mean, yeah, most likely. If we're gonna say that the doses 2 to 3 weeks apart are basic, or 3 to 4 weeks apart, are basically like getting a single, initial prime, and then this 6 month thing is the booster. Then yes, I mean that's basically what we're saying.
Brad is an Amy fan here. Look, you're a major reason I listen. Don't listen to the haters. I don't. I got my first Pfizer jabs 10 weeks apart last June. Booster, yes or no?
June, booster no earlier than January. Push it out as long as you can. I actually got my second shot at the beginning of last January. So yeah, I would say no earlier than January. Push it out as long as you can.
Howard Dixon's parasites discarded. They weren't. They're still sitting on the bench.
So if anybody wants them, you have to either get in a car and pick them up, or you have to send me and Vincent detailed instructions of how we would ship containers full of formalin, because I don't think you can. But if you can figure it out, we'll be more than happy to get them to you.
Yeah, the best thing
They're
is if you're
creeping
local.
up my undergrad.
If you're her undergrad who wants to be a doctor is creeped out by parasites. Can you believe it? Ay, ay, ay. Look, Amy, you are perfect the way you are. Never change. Okay, Amy? Amy gets very upset, so I'm glad you guys are saying this. Yes, got my blue glasses. Check. I'm home. I figured it out. All right. Are disulfide bonds on viral surface proteins less antigenic than other amino acid residues or secondary structure motifs? So the disulfides, Amy, are they part of epitopes?
They can be.
I don't see why not. But if they're involved in a disulfide, would you think they would be buried somewhat or not necessarily?
Not necessarily. I don't think all the disulfides bonds of the capsid of a picorna are buried. I think that's what makes up the turns, right? Of the
Not
beta
sure
sheet.
yeah, but I don't think there are disulfides in them. I have the sense that they're buried, but I think we just don't know.
I don't know.
Carlos wants to know what metrics will tell you when it's safe to go out without a mask indoors.
Safe to go. I think when there's when we're at the level of 1 case per 100,000.
What are we at in like New York City? Do you know?
Well, we had been, I think, hovering around 8, but I think now it's like 16. Don't quote me. I'm not 100% sure. Okay. And I heard Scott Gottlieb, and he thinks that vaccines for people under 5 will be out at the beginning of next year, by February, the latest.
Excellent. I'm scared to get vaccinated. Can you give some words of encouragement? Well, the two people you see here, we got flu vaccine last week. We got third mRNA this week. I ran out. Amy got a vaccine in December of 2020, early. So, and all, our families, Amy got her dad vaccinated. Her mom is vaccinated. My family is vaccinated.
And my family has my family has health issues, severe health issues.
Yeah, so
right?
our loved ones are vaccinated, and we are 100% confident in that. I don't know what else I could tell you than that. I have full confidence in them. And if you want to know more, ask questions, because I think that's the best way to get over fear, is to find out what is scaring you and then see if you can get a good explanation. I'm happy to help.
Yep.
Yes, we both got a third dose. There you go. We saw the data. And the University of Hong Kong preprint we just discussed, but Amy has issues with the data, which I fully agree with. So we'll see what they look like if they're ever published.
Yeah, I don't like this I don't like this idea of 70-fold faster and 10-fold lower because if you look at the slope of the line, that's not what the slope of the line says. And rates have to be defined. So this is a rate. It has to be defined by the slope of the line. And that's not what the slope of the line says. So go back, do some math, recalculate, get out the big button calculator, try again.
Amy, Vinny had a great day. He gave a lecture on virus evolution. One of my favorites, yes. It was a very nice lecture. Great questions. Thank you.
Great.
Ontario is moving to boosters 3 months after the second dose. Seems like this is too early. What are your thoughts?
Too early.
Yes. Too early.
Too
I
early,
agree.
making the same mistake. Too early. Try 6 to 9 months afterwards. No earlier than 6.
The data from Canada say 6 months. And so if it's not tested shorter, then at least 6 months. Are there specialists that focus exclusively on emerging, evolving viruses as opposed to specializing in certain viruses? Of course, yes. There are people who study emerging viruses of various sorts. Would you say Ian is a specialist in emerging viruses, Amy? Ian Lipkin?
Sure. Yeah, I know who Ian is. Yeah, for sure.
And then there are people who specialize in the genomics of emerging viruses. So yes, there are lots. My brother believes he was one of the first people in Canada caught COVID back in January 2020. He's had 2 vaccines. Is it possible to test to see if he was infected back then?
Sure. Go look for nucleocapsid antibodies.
Yep. And I think some of the tests do look for nucleocapsid antibodies, right, Amy?
Those are all the antigen tests, but he might be beneath the level of detection for that. I don't know what the level of detection is for that, but I'm sure that when you go to a laboratory, not the home kit, that there's a more sensitive test. Like the home kit is like you did the pregnancy test, but you always go back to the OBGYN to just confirm it with a blood test that you're actually pregnant.
Oh, and we might be able to do the things we love again by 2030 or so. Bill Hazeltine. Let's not talk about Bill. Let's not talk about Bill.
Yeah, it just gets us aggravated.
There's another one who's, what do you call it? Scaring
people. He moved to the dark side.
He moved to the dark side. Thank you.
He's a scaremonger. Is
it
it
drives
too
me
early to go on vacation for Christmas? Oh, wait, I'm Jewish.
You're Jewish. You can't.
Yeah, I just realized that. Not going to work out well. Okay.
It drives me crazy when I hear people quoting obscene vaccine death counts from people citing VAERS. Is there a better accounting of actual vaccine adverse events? Right, so VAERS is basically the raw data. People write in, oh, my uncle got the vaccine and he died a month later in a car crash. So...
Do they even say a month later in a car crash, or do they just say he died a month later?
That's a good question. So basically, CDC goes through all of these, and those data are available. I just don't know where. I'm sure you can find them. They go through and say none of these deaths are related to the vaccine periodically. So, yes, but I'm not sure where. Do you know, Amy?
I don't know, but I heard a clinical neurologist here at Columbia gave a talk saying that death by the vaccine was equivalent to death by COVID for those under 50, and therefore there was no need for a vaccine. And when I went up to Rod's lab, we were all just flabbergasted by that. And we were like, really? We didn't understand what drugs he was smoking and why he wasn't sharing any of them. But yeah. But then when you look at the New York Times website and the Hopkins database does not support his conclusion. So we're just not really clear.
If prior Delta infection doesn't stop Omicron well, does it imply Omicron won't stop Delta well? Weak cross immunity.
No.
Could they then co-circulate instead of Delta being displaced?
Doesn't say anything. Doesn't say that the converse is true, that Omicron will be unable to prevent Delta infection.
I think the idea that Delta infection doesn't stop Omicron is premature, don't you?
Yeah. I think that the whole thing is premature. And as I said on Twiv, he shot himself in the foot after saying X and then coming back with Y, which made no sense. But the person who he was talking to is not a virologist or an immunologist, so wouldn't have caught it. So, yeah, I'm holding out. I don't want to make any statements because the data is not good data.
So Les says, Sean Crotty talks about a VAERS example, which is the vaccine turned me into the Hulk. So you can actually report something like that.
Really?
On the VAERS site.
So what is the Hulk? Like he became green?
Yeah.
Like the Green Giant?
Well, it's different. He's a green guy who, he turns from a human into this big muscular green guy, and he has a temper, but sometimes he does good things. I'm not. Don't ask me about comic book stuff and movie. I don't know.
I don't know, but I could be the next great pastry chef, like Pierre Hermé.
Oh, Amy is a very good pastry chef. She gave us a little box of cookies.
And what were my testers?
Oh my gosh, they're so good. You guys cannot believe how good a pastry chef she is. I told her maybe we should make that the second half of Microbe TV, but that would be a for-profit. That's no good, right? We could sell cookies to raise money.
They just have to pay enough to buy the ingredients.
We could sell them for raising money.
Sure.
What is it that you made that cost $50 at Saks or whatever it is?
The Panettone. Panettone, yeah.
All right, I don't know the answer to this. Frank, can a doctor's office in New York report rapid tests to the state health department? I have no idea. Do you know, Amy?
Yeah, they can.
They can?
Yeah.
But don't they have to be validated with a PCR?
Yeah, they have to be validated. Generally, they're validated with a PCR. But yeah.
Yeah. Mario wants to know if you make virus-shaped pastries.
Sure, we can.
I know people are obsessed with ivermectin. Is it dangerous? Should I try to persuade them not to take it?
Yes, you should try to persuade them not to take it. It is dangerous. You can get neurotoxicity. It's not a good plan. Especially
if people are taking the veterinary ivermectin, which is potentially a higher dose than you should give to people. And there have been many problems with people taking ivermectin. They have to be hospitalized and so forth. So you should dissuade them from doing that. There are better ways to take care of COVID. How would it benefit the world if someone found Bat Zero?
There's no such thing as Bat Zero.
Well, yeah, that's true. It's going to be hard, but I love the name Bat Zero. It's a movie, Amy. Can we make it?
For sure.
Bat Zero?
For
and
sure.
You can be the person who does wildlife sampling and stays in a tent collecting bats. How's that? It's just up your alley, right? I'm joking because Amy said I'll never camp. The only camping I do is in a hotel.
The 3-star hotel. You have to qualify it. It's not a hotel. It's only at the 3-star hotel. I don't go beneath the 3-star hotel. Otherwise, you just get back in your car. You keep driving until you can't drive no more.
So, Fauzi, let me tell you what happened with SARS-1. They found a cave in the countryside where the bats harbored different coronaviruses, all of which, when put together, could give you SARS-CoV-1. So they didn't find SARS-CoV-1, but they found all the pieces that could come together by recombination. So that told us that there's enormous diversity that could form viruses that infect people. So it was an important finding, and I think we would like to do something similar, whether it's a lot of pieces in various bats or maybe in one cave there's something.
I think like SARS-CoV-1 was not considered a recombinant of 5 individual viruses. I think it was thought of to be a recombinant of 2.
Recombinant of what?
2 coronas.
Which one? I'm sorry.
Different. So the
two?
one
Okay.
was 2, where this is clearly 5 or more.
Yeah, at least 5,
according to the Eddie Holmes, Andy, whatever, and even Raul Ramadan, whatever the guy's name is here. It's at least 5. So it would be really hard for me to believe that you could find that in one geographical location. And I think by the papers that we've done on TWIV that talk about the 16 amino acids in the Laos. Yeah.
This is Raul's book. He's a professor at Columbia. He wrote this in a couple of months early on, right?
Yeah.
Been on TWIV a couple times.
But I think like the fact that the 16 amino acids in spike are found in the Laos ones or the Cambodia viruses, and then some other part, the furin cleavage site is, or whatever, is found in some other area. I don't think you're going to have a bat 0 or cave 0. I think you're going to have it. I think what you're finding is how it traveled, right? It went from Cambodia or Laos, and then, I don't know, either via the bat or via the automobile, went to someplace else, did some stuff, then via the bat, via the automobile, went someplace else, did some stuff.
I think it's important to learn this, Fozzie, so that we can not repeat it, because if it was sold, if it somehow got to a live market, we should stop that practice, right? Because that's obviously now twice that this has caused problems.
That's not going to happen. It's a cultural thing that we don't understand, and I don't think we should implement our culture on somebody else. I think what it tells you is that you need to have continuous testing, and you need to have better prevention programs.
Yeah, I agree with you, but does it make sense to have a practice that causes pandemics and a lot of global suffering?
You're not going to get the people whose this is their tradition and this is their lifestyle to change. That's just not going to happen.
Okay. All right. I've heard many people, including Fauci, say vitamin D and zinc may help. I don't know of the evidence. I think vitamin D is not bad to take, especially for people who are not in the sun very much. Amy, you're not in the sun a lot, are you?
No, I don't know.
Do you take vitamin D?
Yeah, I take vitamin D.
Yeah, I do too.
But I don't take it because of this. I take it because I don't want to get osteoporosis.
Oh.
I don't take it because of this.
Well, I take it to just be healthy for all the things vitamin D does. I'm not sure there's great evidence, direct evidence for an effect on COVID, just indirect, which is not convincing.
And zinc? Zinc is the ubiquitous respiratory thing.
Antiviral, yeah.
I
don't think that's anything. How many more variants do you think you can take before you become gibbering Rex? Oh, I don't think it's they could throw as many at us as they want, because they give us a little break in between. You know, there was a nice rest from Delta, right? Things seem to calm down. But I'm sure there'll be more. Don't you think, Amy?
Yeah, I'm sure there'll be more. I'm just not sure that what we think are, what we've deemed are the important alterations are really the important alterations. I think I've seen several papers now that talk about the spike changes really weren't the important changes. And so, yeah, I think it's fine. I mean, viruses are going to evolve no matter what, so it's fine. And then there's going to be a limit where it's on the border of extinction. It's fine.
Callista, thank you for your contribution. Callista sent us face masks that she made early on.
Yeah, we love her face masks.
They were great. Beautiful. And I'm coming out to LA next year. You're going to cut my hair, right? You promised to do that. I'll come back, Amy, with a crazy hairdo, you know. Look at that.
I'm looking for new color. I think this old color, you need to spice it up.
What color do you want?
You could do, I don't know.
We'll let Callista decide. And I have to go out to California to do some fundraising next year. So maybe we can have a cup of coffee.
You're going to LA? I thought you were going to San Francisco.
Yeah, both.
Oh, okay. Well,
Jeff is in LA.
Yeah, well, I haven't heard any money from the lab. So I might not be so happy with it, since I don't seem to be seeing any money towards the lab.
Patience.
My February 5th is coming rapidly, and the discount that the company was willing to give me for my $50,000 experiment is expiring on Tuesday. I don't think I have a lot of patience.
All right, so Kang wants to know what prevents antibodies in breast milk from being broken down by the digestive system.
I think that they're also, I think some of them might be like within the lipid droplets of the breast milk. Like milk is really fatty.
Yeah, I think the IgAs also have the secretory piece that protects them, right?
Mm-hmm.
And the others also can be, maybe it's also a quantity effect. You have so much. But
I don't know anything about quantity effects. You might want to talk about that with your host from yesterday.
For Stephanie Langle. Yeah.
No, I meant the other one. The one who said, oh my God, maybe it's just a big bolus of weakly binding antibodies.
Okay. Can virus evolve to avoid lung cells since it doesn't help transmit and only puts the person in bed? Or are upper cells too similar to lung cells for the virus to hit only the upper?
No, they're very, very different. And for many, many years, it was thought that rhinovirus only replicated up in the upper respiratory tract.
So, Amy, in most people who have a common cold caused by rhinovirus, does the virus get into the lower tract?
In 90% of the people, no.
And that's because the immune system stops it?
It's part the immune system. It's also part the way the virus works. It doesn't lyse cells, and it doesn't cause CPE, and it moves more cell to cell. So like when you do modeling that the Palmenberg and Gurn lab with Yuri have done, you just see the cells pop off, and then there's no damage to the underlining. It gets immediately filled in. And so then it's very highly dependent. So Anne's philosophy is severity of disease, is, at least for Rhino C, is very highly dependent on the beating cilia. So the cilia beat and they move the mucus all the way up the upper tract, and then you cough and you digest it. But because Rhino C receptor has something to do with whether or not the cilia beat properly, they're not in coordination or something. She's trying to figure it out. That it doesn't move the mucus, and so that it keeps getting back reinfect. It almost like continuously reinfects the cells.
Anyway, so yes, I think I agree with Amy. You could get a virus that it differentially infects. Yeah, I don't know if
I'm not sure. Yeah, I'm not sure that. Yeah, I'm not sure that also like 90% of EV68 is not just all upper respiratory.
Well, we don't know what fraction of COVID goes into the lung. It's not 100%, right?
No, by far not. It's like 3%.
Will the original antigenic sin be a concern about the call for more boosters? No, I don't think so, because most people have seen the original SARS-CoV-2 virus, and that's immunity against that is okay. It's okay for protecting against all variants. So even if you had original antigenic sin against that, it's fine. It's absolutely fine. Oh, Kate lost her power, but watch on your phone. Yes. What's the rationale for using remdesivir when in the trials used to justify its use? It was the least effective and has side effects. Well, Daniel says he doesn't use it. Now, a study just came out saying if you treat unhospitalized patients, so you're treating them earlier, it gets a certain percentage of them from going to the hospital, which is a positive thing, right? But I think
I'm not
that
sure it
is
cancels out the kidney and cost of it.
no. I
I
agree
mean,
that
I'm not even sure it was as high a number as the 30% efficacy of molnupiravir. And we don't
yep,
think that
yep.
that's very effective.
I agree. I think that the remdesivir shouldn't be
So,
used anymore. But many places use it. Hey, many places use ivermectin and hydrochloroquine, even though they're not recommended.
what can I say?
I got Pfizer 3 months after J&J. I'm coming up to 6. Hey, yeah, you should get a 6-month boost. Yeah, right, Amy?
No, he got his last thing 3 months ago. So he should wait. And he should wait not from the first shot. It's 6 months from the Pfizer shot. So he doesn't go until
Oh,
like
okay.
April.
Yeah, go for 3 more months. Yeah. Sorry. Yep.
No.
I think I perturbed Vincent during the last Q&A by pushing the point that I thought N95s should be worn as much as possible. Don't worry. I really am sorry that I occasionally get perturbed. I shouldn't. I should not be so emotional about science. Well, maybe that's what is good.
Then why do it?
Yeah, well, I shouldn't get perturbed, okay? I'm sorry if I get perturbed. From now on, if I get perturbed, I apologize in advance. So Mark wants to know what kind of things you're working on in the lab. You have a paper just accepted, I hear, Amy.
I do. I got contacted today by Ian's administrative assistant. They want to social media it to death, right? So I go to the website. I'm all excited, and it says manuscript ready for publication. It said that last week. I click every button that you could possibly click. Manuscript ready for publication. I eat some chocolate.
So when it's published online, then you will come on Twiv and talk about it, right?
Yeah. I think even though others, like the people I sent it to in Europe, did not get it. I think it's a big thing.
Yeah, I think it's terrific. Can a single virus target multiple receptors in one or different species? Yeah,
Absolutely.
Sure. Absolutely. We have examples of that. Yeah, for sure. Welcome to the third booster club. Isn't the name of a movie? The Dallas, what was that? The Dallas what club?
Seriously? You're asking me? I don't know, but it's
Dallas Buyers
842.
Club. That was all about AZT, the Dallas Buyers Club.
I don't know. It's
Yeah.
842, so like a few more, and then I have to go.
Okay. Have you seen the latest data on the intranasal vaccine? It provides superior protection against heterologous respiratory challenge via IgA. Is this a SARS-CoV-2? I suppose so.
Yeah.
Everything is about.
Yeah. Go listen to the TWIV. We gave an in-depth explanation.
Did we?
I did.
So this was a while ago.
Rich, no. Just on Friday. Remember, Rich said, "Oh, I don't know what all this neutralization assays mean. If it doesn't mean protection, it doesn't correlate to vaccine effectiveness." And then we talked about how it was all IgG and not IgA, and it's IgA that's really important. Go back and listen to the TWIV. You too, Vincent.
That was respiratory syncytial virus.
No, it was
Friday.
Not. It was in discussion to Omicron data from Pfizer.
Okay. All right. Go back and listen to that.
It was at the very beginning.
So someone says here, discrete entropy doesn't trust any academics who don't get perturbed. LOL. That's true. You're right. But I still feel badly when I get perturbed. Okay, here's the thing that I told Amy about.
What is this?
I found you from Lex Friedman and I have been a big fan. I wonder if you've seen Jamie.
Yeah, this is the guy. This is the guy that I listened to the first 45 minutes of his stuff. And he's just, I mean, Lex eggs him on, and Lex basically says, I believe that there is no natural host. I believe that and then I forget. It was just, yeah. And then
All right. That's
he
just
like
unfortunate.
tweeted with Daniel, and Daniel responded to him actually.
Yeah, he sent an
That
email to Daniel.
yeah, and so he's like some evolutionary biologist or something. He sits and then he like talks about like gain of function, and he misdefines it, and he says that um because you can't because um the Bat Lady and Wuhan wrote the Eco Health Alliance grant in
Yeah.
2018 where they were going to introduce the fear and cleavage sites, and it didn't get funded. And so you absolutely know like Barrick's lab did not do those experiments. Although I do believe that he did something quite similar. Um that she probably did those experiments even though she claims she did not. I believe. He says by absolute means she did those experiments, and I'm like sitting there, well, where's the fucking paper? Because that would be a huge paper.
Well, I'm very disappointed in Lex. I'm going to email him and tell him that. Not that he cares, but
I mean, the whole thing was a travesty, and it's just one big bullshit artist about bullshit artist, and
too bad.
And it was
Well,
like,
I'm
and
sorry.
he talked about like how he then quotes about like how he had Collins on to talk about something or other, and they talked about the natural origin hypothesis, but he never like can piece together that you were on it to discuss it and knew a more. And then they talk about like they implicate David also into the saying that the fear and cleavage was a smoking gun, which yes he did
later.
say, and
okay.
Well, he kind of takes back, but
right,
whatever.
Anyway, it's too bad.
Yeah.
I'm very sorry, but Stephen, at least he had one good guest. Probably others. Callista has a headache too. All right, so that's what you have, Amy. I'm sure.
Sure, we can go with that.
What about AstraZeneca at 12 weeks, 4 months?
Yeah.
I think it's okay, but I think 6. The data say 6, right? I think there were data. Oh, you're right. There were data initially at 12 weeks. So
yeah, I
okay.
think 12 weeks is the
fine.
minimum for AstraZeneca, but it's a different platform. It's a different type. It works slightly differently because it's virus.
James is waiting for his 7th booster. It's his lucky number. Yeah, you go. There you go. This is it. No, just 3. I don't think you need more. I would really resist that. Thank you, Jiching, for your contribution. I really appreciate it. I got 2 shots of Janssen. Could I get an mRNA in addition? You could, sure. There's no downside. Mix it and match. Mix your vodka and scotch and beer. No, I'm sorry. Don't do that. You can mix up your shots.
Why don't you just tell them to get a hangover?
No. I'm sorry. I apologize. You could do it, yeah. Although the recommendation in the U.S. is 1, because Janssen is 1, right? And so get a second Janssen.
2.
After
so
2.
Johnson is not 2. All right,
Janssen,
2 more
it's
questions.
the same thing.
Yeah, but Janssen is the Johnson & Johnson.
I know it's 1. It was originally 1 shot.
Yeah, but now it's 2.
So now you can get a second. I don't think you need a third, right?
No, they're saying get a third.
Even if you space the first 2 properly?
You want them to actually do some trials and analyze some data.
All right,
that's like
Mark,
holding up
there's
the
no
envelope.
problem. You can mix them up. I always appreciate Amy. She takes time away from her work and life to help us. She looks tired many times. I hope she takes good care of herself. Do you take good care of yourself? She's not going to answer it.
That's like ridiculousness. All right, 2 more questions because it's.
I'm sick today because I got a booster and a flu shot at the same time. Same arm or 2 different arms at least. Yeah, you're going to be a little sick, but you'll get better.
Why would you? I did the same arm. That's probably why my arm doesn't work so well.
I did separate arms. What is
I
the
told
validity?
you earlier that my philosophy may have been flawed.
Yeah, you agreed. I agree. What is the validity regarding Omicron infecting fully vaccinated people easier? As we said earlier, I don't think those data are good yet. It's too small. It's too early to tell. All of these preliminary early findings eventually get overturned. So just wait for more. Mark, here's gives $5 for some Amy's. You get some Tylenol, okay?
Sure.
Miter wants to know, what is somatic hypermutation? Do you want to take a crack?
No.
So antibodies come out of the bone marrow. B cells come out of the bone marrow. They go into the lymph nodes, and then when they recognize an antigen, they proliferate and they make antibodies. Those early antibodies don't bind the antigen, the protein, very tightly. So in the lymph node, there are these centers made, germinal centers get established once you have a virus infection. And there, the genes for the antibodies that are in the B cells get mutated. There's an enzyme in the cell that mutates them and randomly makes antibodies with different specificities, and they test them against the antigen, and the higher affinity antibodies get kept. Those B cells get kept. That's affinity maturation by somatic hypermutation, because it's getting, the DNA is being mutated in a somatic cell. It's not a germline cell. So that's a brief explanation, and that's why you need time between boosts to allow somatic hypermutation to occur. Is that good, Amy?
Yeah, it's excellent. All right, two more and then I'm going.
Can you talk about the antivirals? So molnupiravir and Paxlovid, Amy, do we know anything new?
No.
One has a EUA, right?
Yeah, molnupiravir has an EUA.
EUA.
And it's like 30% effective, and it's a polymerase targeting drug. And Paxil is going up for EUA, and it is a protease inhibitor.
We've talked about them a lot, Thomas, on Twiv, so you might want to go listen to those episodes. Mark wants to tell you how grateful that he is for you. Grant, thank you for your contribution. Mario, thank you for your contribution. From Canada, who should get the third shot? Who shouldn't? Some people got their first and second a few months apart. What do you think about that? If someone got 2 shots 3 months apart, would that be okay? We'd be guessing.
Sex.
We'd be guessing.
I'm guessing, but I prefer sex.
So if you haven't got 6 between the first and second, I would say get a third shot.
Yeah,
right, Amy?
I mean, yeah, because the reality of it is you give the polio vaccines every other month for in the first 6 months, because they're trivalent, or they were trivalent, and you didn't get equal immune response, or not all of the serotypes were equally immunogenic, right?
Yeah.
And then you got a big boost at 1 year, and at 2 years, and then at 5 years. So I don't think that 3 months, a few months apart, I'm not really clear what a few months is, but I don't think 2 months or 3 months is sufficient.
Is there concern about original antigenic sin with SARS-CoV-2 vaccination, or even antibody-dependent enhancement, especially with multiple boosters? You don't have any concerns with them?
No.
Yeah, the vaccines seem to be fine with the ancestral strain because of somatic hypermutation, which diversifies and broadens their reach. So I don't think that's a problem.
And I don't know I know about Paul and Theodora, but still, I don't know. I don't know what Paul and Theodora.
Well, they say that it's
ancestral, very confusing to me, because they say one thing, they write another thing, then they say something else.
The vaccine, when spaced properly, can recognize different variants. Do you expect a fourth dose will be recommended for immune-compromised individuals?
Hopefully
What do you think? We're not sure at this point. But, immune-compromised don't even often do not respond after 3. So that could very well be a special population.
No, I don't. I think after three strikes are out. Give them there's no need for them to for you to keep vaccinating them. The way to treat them is to give them maintenance of the long-lasting GSK monoclonal antibody. That's what you do. The others is just ridiculous.
Thank you, Calix Yukon, for your contribution for the incubator. You're welcome.
All right, one more and then I have to go.
Okay. Should you get a third shot if you got COVID about 6 months after the second dose?
You don't need a third shot.
I think the combination of infection and vaccination is also very good.
Yes. I saw that in a paper that I think I sent you and Daniel.
Yeah. Thank you very much, Amy.
All right. I'll see you tomorrow. We have CO2s. We have a lot of CO2s to change.
All right. Just remind me so I don't leave and forget and have to come back. Oh, well, she left. Okay. All right. Now, behind me is the glass virus, but also I have to move away from the mic. This is polio virus. So in the middle, it's polio in the capsid proteins. This is painted by Eric, who used to come here. He's a PhD student in Philadelphia. All right, let us see here. How exactly does the boost help with T cells? Thank you, Real Qualia. That's a great question. The T cell epitopes, in contrast to the B cell epitopes, do not substantially change in the various variants. And the same with Omicron. So we expect the T cell response to be the same. So the third dose, what is it going to do to that? Well, that's a good question. Is it going to broaden it? It's already good enough. I don't think it's going to be worse, if that's what you're worrying. Wouldn't we have more complete protection if exposed after the 2 doses? Well, that may very well be, but you can't be. But I do think the T cells would prevent hospitalization and death. The only issue is, let's say you have the 2-dose vaccine schedules closely spaced, so your antibodies do not neutralize Omicron well. And so now, down the road, you get exposed to SARS-CoV-2. It takes a few days to get your memory B cells making antibodies. In that time, the virus is reproducing. Now, and once those antibodies are made, it doesn't do a good job of neutralizing Omicron, so maybe the infection proceeds a little more than it would have with Delta, say, because Delta could be neutralized by your 2-dose antibodies. And then the T cells kick in, but maybe with Omicron, the infection's gone a little more, so you may have more severe disease. That's why I think the third dose is a good idea. Maybe you do need to have some help from the antibodies before the T cells kick in. Hope that makes sense. I have a friend that had a day or 2 of downtime after receiving J&J. Would it work for their booster to be one of the mRNAs? Yeah, you could mix and match for sure. There's no problem with that. It's all okay. There's no evidence to the contrary. Would you have less of a reaction? Very hard to say. I had, after my second Moderna, a prolonged soreness and a fever for a few hours. No big deal, but and then I got a Pfizer yesterday. Nothing, just sore arm for a couple of hours. Is it still sore? Mildly sore. The other shoulder, flu vaccine on Friday, nothing. So, and those 2 are mRNA vaccines anyway, so it's not even worth comparing. I doubt science will ever find a definitive answer for long-haul COVID. Too heterogeneous, still no answers for EBV reactivation. So don't be negative, overjoyed, because Ian Lipkin is making very interesting inroads on chronic fatigue, ME. Interesting signatures, metabolic signatures. So I wouldn't be so sure. Please be more hopeful, okay? I understand your hesitance, but I think there's reason to be optimistic. Much talk about this faster spread of Omicron. Does it mean spreading to more people faster or time of exposure to symptoms shorter? So that's the thing, Breestar. We don't know. Now, fast spread is not really, just by observing this variant taken over, it looks like it's moving quickly, right? But in fact, it's just displacing Delta, which probably would have done the same thing. It would have caused outbreaks because these are mainly in unvaccinated people. Yeah, the virus can infect vaccinated people, but they generally don't get very sick. And so, yes, I think the messaging about fast spreading is really wrong because all they're doing is seeing this displaced Delta. It's not spreading any faster. They cannot control for an outbreak. They cannot say this would be any different than Delta. I'm sorry. They think they can, but they cannot. So we may eventually find some reasons why it's more fit. So for Delta, for example, Well, there's some evidence that it might reproduce quicker, but even that hasn't held up in the laboratory. So right now, I would say, and those graphs that I showed you earlier of the Hong Kong study where they said it reproduced faster in bronchial epithelium, not even clear that that's right. That needs to be looked at more carefully. So I think it's way too early. Don't worry about the messaging. One thing you should understand from me and here and Amy, Twiv, the greater messaging is often misguided because it's hysterical, and that's what news outlets need, right, to get attention. I don't need to get attention. I'm teaching the world. If you want to listen, that's fine. You know what? If you don't, that's your choice. I'm not going to say something to get your attention. So we tell you the way it is and try and calm you down. So forget the messaging. If you have an issue, come here on a Wednesday night and we'll tell you why the messaging is off. All this baloney about, huge overloads of this and that, it's just baloney. If you're vaccinated, you're going to be okay. Will Omicron be a good thing? Less morbidity, spreads fast. All right, so those are two things we don't know. Less morbidity? No, I don't believe it. Too soon to tell. I mean, this is wishful thinking. This is finding in the data what you want to find. I'm sorry. Spreads fast? All it's doing is displacing Delta. I mean, let me think about this another way and just use common sense logic. When SARS-CoV-2 was first discovered, it was spreading pretty well among people. You really mean to tell me that every variant is getting better and better? No, it is not. They're just displacing each other. Whether there's any intrinsic difference in spread to the virus, which we call transmission, who knows? Who knows? So, Slinky, if those two things were true, then it could be good, yeah. I have serious doubts about whether they're true. At least we just don't know. We just don't know yet. So yes, Jeff, the data drove me to get a booster. Everyone is telling me you should get it, but I didn't see the data. That's what drives me. I never said to anyone, don't get a booster. You make your own decision. And now last week I saw the data and I said to Amy, I think this is good. She said, yeah, I agree. So we went at Columbia and got it. And so far I haven't seen data about, the variants and certain aspects of them, so I'm not there yet. But as you see, when I get the data, I do change. Mother-in-law visiting after going to a party the day before. She will come unmasked but has 2 Pfizer. Should I ask for PCR same day? 2 vaxxed, unmasked. So you could do a rapid antigen. I would say that's going to be easier than doing a PCR. And it won't be as sensitive as the PCR. The PCR may be positive, but it's not really an issue. It's not really an infection, for example. So do a rapid antigen and see what that says. And if it's positive, then you should go get a confirmatory PCR. And then, if the CT value is low, then she should stay away at least for a couple of days, right? But I think the 10-day quarantine is in fully vaccinated people is not really needed. Personal opinion. What are your thoughts on the universities closing? Well, I think this is absurd. Frankly, the kids are all vaccinated. A lot of these universities are random testing, so they pick up positives. The kids don't have symptoms. What is the point? They're all vaccinated. What's going to happen? They're going to spread it to each other? So if they do, what's the difference? They're hardly going to get sick. Are you worried about spreading it to unvaccinated people? Well, that's their concern, not yours. So I think these are terribly misguided. And whoever is advising the universities is simply wrong. We have vaccines, folks, that work. No more shutdowns. Come on. The viruses are not evading the vaccines. They still protect you against serious disease, and that's what we care about. If the vaccinated don't transmit, how did Omicron spread in Australia when only vaccinated people can go there? And that's first, all right, they can only go to Australia. And how are there hundreds of cases in Gibraltar when it has a 100% vaccination rate? I didn't say vaccinated people don't transmit. They can, at a lower rate because they have certainly less infectious virus than unvaccinated people, so their likelihood of transmitting is low. I mean, already, in fact, non-vaccinated people do not transmit very well. 80% of transmissions is by 20% of the infected people who have the highest viral loads. So that's going to be even less with vaccinated people. When only vaccinated people can go there. So a vaccinated person probably brought it in. Low levels of virus, or maybe that person who was vaccinated, it didn't take in them, right? You're assuming that everyone who's vaccinated makes an immune response. It's not the case. The vaccines are not 100% effective. So if you vaccinate 100 people, there are going to be 10 of them who might not have made a response and who can transmit. So think about this, Adrian, before you use that logic to indict the vaccines. It's people's responses to them. And that's, remember, vaccines are not 100%. And so those 10% can be the spreaders. This is an interesting comment. A virus has a mutual agreement with our immune system. It will not risk sharing corrupt files. That's very interesting. If stupidity in the world causes you stress, you're doomed. Yes, so it does stress Amy out. We talk about it every day. And yes, she's just doomed because it's not going to end. I'm double jabbed. I go on holiday in February. We'll be amongst more people than normal. Does it make sense to get the booster a week or two earlier? Yeah, just get 6 months between your last jab, okay? But 2 weeks, 2 to 3 weeks before, that would be good. You would have your maximum antibody production, yeah. Are you excited about the plant-based vaccine from Canada? I've been excited about plant-based vaccines for years. I've been talking about them in the course for a long time. I think they're very exciting because they can be made rapidly and cheaply. Unfortunately, the influenza plant-based vaccine isn't much better than the egg-based or the cell-grown, but maybe SARS-CoV-2 will be better. That would be really nice. Christian, but Jostin back for another TWIV? Okay, yeah, that's a possibility. I think towards the end, I'd like to have Barrick back to summarize everything, right? And here's Rogan having bad people on. Look, they don't want me on because my Friedman episode was less than 1 million views, and I'm sure this guy who was just on is going to be more than 1 million because he talks about what people want to hear. I mean, you guys here don't want to hear that nonsense. It's obvious. But Lex's people, Rogan's people do, so that's why. And, you know, Lex had a moment of weakness having me on. Not happening again. What's the selective pressure driving change of the virus? Okay, so this is a good one. We talked a little bit about this in our evolution course today. You folks know I'm doing a live stream virology course on YouTube right here. Well, it's almost over, but the videos are there. At every reproduction cycle in a cell, a virus gets in, it mutates. It makes lots of mutations in its genome just randomly. And then those go out into the world and see what happens. Now, SARS-CoV-2 is no different. And what we're seeing is a series of variants being selected and being more fit than the previous one and therefore dominating in the population. Why they are more fit is not known by anyone on this planet. And if someone tells you they know, they're lying because the data ain't there, folks. It could be immunological fitness. It could be like influenza virus. Variants arise each year that evade antibodies very slightly, and that's enough to give them a fitness advantage. It doesn't have to be a complete block. So now we have SARS-CoV-2 variants emerging. What is the selective force? Is it better binding to a receptor? No, just because it's better doesn't mean that's it. It could be these immunological changes, but not proven. Need to do some experiments to sort that out. Does it reproduce faster? I don't think the data are in yet. For Delta, certainly not. And Omicron, the data we showed you are not convincing. Anyway, so that's the problem. We don't know what the selective force is. And don't let anyone tell you that they know. They can hypothesize, and that's the basis of science, right? But they do not know. They do not know. Yes, we passed 100,000 on YouTube a while ago. Now we have 103,000. I think it's great. I'm happy that so many people want to learn virology. I hope many more people come. I mean, I plan to do more and more continuous work at the incubator, not only on these podcasts, but on other things as well. So hopefully we'll get more and more. Yeah, someone on Virology Live commented that misinformation is the virus with the shortest incubation time. Yes, I think that's a great one. That's something we have to save. I had one dose of Pfizer, but the second was J&J after bad reaction to the first Pfizer dose. Am I still good? So it depends on the spacing apart, right? Was it more than a couple of weeks? If not, then you should get a third dose. If it was 2 months or more, then I think you're good. Thank you, Pam, for your support. Really appreciate it. I reckon he specifically wants people who will go against the status quo so he can generate clicks. Yeah, that's fine. He can have his model. The thing that bothers me is this is science, and I don't think you should mess with science in that way. You shouldn't use it for clicks, but they are. And every time there's an outbreak, this is what happens. And we don't do that. We just get good scientists on the show, and as a consequence, we get fewer viewers. But the viewers who come, they appreciate it, and they learn a lot, right? Do T cells go through some kind of optimization over time as well, like B cells, somatic hypermutation? Good question. I know that, of course, they have variable antigen receptors that are assembled by recombination, like the B cell receptor is. I don't know. Good question. That's a hole in my immunological repertoire. Let's see. Does anyone know? Put it in the chat. I got the concurrent chat running here so I can check it out. I theorize you don't need selection to cause a more transmissible but less virulent. Plain random chance. Yeah, but we haven't seen that with any other virus, Steve, right? And we've been looking for a long time. Yeah, I agree, but we just haven't seen it. Now, maybe we see it with Omicron, but it's too early to conclude. When we find the data, I will agree if it warrants it, but it's going to be some time. Somewhere on Twiv, there has been a study that correlates neutralizing antibodies 54 days after vaccination. No, I don't remember. Does anyone remember that? Anybody remember? Put it in the chat. Okay, here's the answer. Somatic hypermutation does not occur in T-cell receptor genes. So the variability of the regions in the T-cell receptor is limited to the germline segments. Thank you, Dr. Google. The last 11 weeks, we've had 100,000 U.S. deaths, fastest other than the 2020, all in 94% in unvaccinated people. The virus finds them. Boy, does it find them. I don't know. No, I don't know anything about the Sudan thing. I have spent the day immersed in viruses. If I have had AstraZeneca 12 weeks apart, 4 months, T-cell wise, you're good to go. If you and so 4 months is the gray zone, right? I don't know. We got 6 months for sure. It's probably slightly less at 4 months. So it's really a gamble, Mark. I don't have any data to advise you on. After TWIV 841, I took Amy's suggestion and started writing the Amy is Right pandemic. That's really funny. Do you want to write the preface? If you ask me to write anything, it'll be late. I still owe a chapter to Fields of Virology. I got to do that this week. Oh my gosh. So to bring this up, do T cells go through somatic hypermutation? No. Germline V regions of T cell receptor is what you get. Yeah. Alex Siegel said estimated efficacy against severe disease is 95%. Blah. This is BS. There's no formula for predicting that from neutralization, because the protection against severe disease is done by T cells. So how the hell would a neutralizing antibody titer tell you anything about that? I need a sip of water. Someone said when I sip my water, I have to show the TWIV logo as branding, right? Could SARS-CoV-2 Omicron hide in adipose cells? I don't see why it couldn't. We've been surprised that virus is hiding out in various places. So it could, but until we see it, we don't know. I was in the Moderna trial. Second dose January, August, got booster a year later. 1 year is good. According to the data, you should be fine. What does it mean to say Omicron is more infectious? Well, that's a good question, Sally, which you should ask the next CNN reporter who says that, because all they're doing is repeating what Peter Hotez said. I don't know what it means, because they have to define it. So does it mean that 10 viruses will infect you rather than 10,000? Or does it mean that it reproduces faster when it gets in? Ask them to explain it. They'll probably say, oh, it spreads better in the population. And that to me means it's more fit. Yeah, it could be that an individual is contagious longer. That's a great explanation for more infectious. But they don't know. They're just throwing out this word because it scares people. Good question, Sally. Very good. Ontario is allowing booster doses after 3 months. Should we wait? So I would wait, if you could, to 6 if you're in an area that doesn't have a lot of activity, or if you can restrict your exposure, I would wait. Yeah. Is there any virus that modifies the cytoplasm to make it more viscous? That's a good, interesting. The reason I like teaching is because every time I get questions I haven't heard before. So that's an interesting one. And no, I'm not aware of any. It's already pretty viscous, and you need to use motors to move around in that viscous lake. So I'm not sure that would be good for the virus, because the virus particles have to move around too. Thank you, Mark, for your contribution. I think I answered your question. Yeah. Amy's gone, but you want to know about Laura Walker. So I had Laura Walker on TWIV in 2019, 2018. I thought she was good. She's a bit glib. But I don't know what she's done lately. And I think she's said some things that would be not pleasing to Amy, if you know what I mean. Rima, please, any of you, let us know when you visit New York. You can visit the incubator, but I will be in your neck of the woods next year in Nebraska, and I'm going to let you guys know what my schedule is. I just don't have it yet. I'm sorry. Did you say in the last TWIV that most of the South Africans diagnosed with Omicron had previous infection? I think that's what Daniel said, and he gave a reference for that, so you should go look that up. I'm a bit confused. Got a dose of Pfizer in August, just got my second. Do I need a third? So the idea here is that the third dose corrects the too close spacing of the first two. But we don't have an experiment where people just got 2 doses 6 months apart, right? So I'm hesitant to say you're okay. It wouldn't hurt to get a third, just to be sure. Theoretically, you should be okay, but the data aren't there to say that, if you know what I mean. I work in a hospital, wear a surgical mask, which I switched to an N95. I think you should wear an N95 from the beginning of the outbreak because you get a lot of exposure. And an N95 is better filtration for that. But not because of Omicron, no. If you haven't been wearing one up till now, then don't switch, because Omicron does not change the masking. You think a vaccine that matches Omicron would be helpful? No, I don't think you need it, because the ancestral vaccine is working, especially if you got the booster properly, if you just do the dose spacing, and the T cells are induced by it. So Omicron would just reset, right? You'd now have people immunized with Omicron, and you'd have different variants arising as a consequence. So I don't think that's a good idea. Thank you, DR, for your contribution. April 5th, JJ, Moderna, November. Realistically, what's my status? JJ, really one dose. It was a quick hit. Also, can you do a TWIV or immune on JC and natalizumab? Okay, we can do that. As I said, 2 doses correctly spaced should be sufficient, but we don't actually have the data for it, so that's my only hesitation. If a booster, say 5 months, does the job, are you protected from Omicron in the first 5 months before antibodies? So it's a gradual thing, right? It's not overnight. So you have, as the antibodies mature, you have an increase in protection. Thank you, Cassie, for your contribution. Vinay Prasad. I don't know who that is. Let's see. Okay. Well, about vaccines. It's important to have vaccine equity, which is absolutely true. So Vinay has said that, and that's a good thing to say. But I don't know anything else about him. We're approaching 1,000 people. Look at that. Can we break 1,000? Can we get more likes? Thumbs up, man. With 900 people, we should have more. You know, we're little peanuts here, so we might as well get as many thumbs up as we can. I didn't have a choice of brands, so I had 2 Modernas originally, and then I asked for Moderna. They said, we don't have any. I said, so give me Pfizer. It's fine. Doesn't matter. What's scaring me is having out-of-town visitors fly in. I'm having them stay one night and rapid test. They're triple vaxxed. Oh, yes. Rapid test, triple vaxxed. You're good. Don't be scared. You know, folks, at some point we have to come out of our caves, right? And the vaccines can let us do that. What would happen if lipid droplets with sulfur in them attached to S1 of SARS-CoV-2? Well, it would be large. It would probably block attachment, right? Because the droplets, depending on the size of the droplets, I would think they would be very big. My mother-in-law is afraid of long-term side effects from the vaccine. Many people are, and we don't know of any because it hasn't been long-term. But as Paul Offit said, and he's an expert on vaccines, there usually aren't any long-term effects. You know, the rare side effects, the clotting with the adenovirus vector vaccines that we observed, we picked up pretty quickly as soon as it got into many, many millions of people because it was a very low-frequency event. It wasn't a long-term event. So I would say that she shouldn't be afraid because, well, first of all, if it's long-term, what are we talking about, 40, 50 years? Really? Is she going to be like 90 or 80 and worried about that? I think you should just live your life and enjoy it and don't worry about it. You're not going to have long-term side effects. Well, no one can say you're not, right? You can't say no. But you just look at the data we have. It's really unlikely. So I don't think she should worry. If an Omicron-specific boost is available in March, like Pfizer says is possible, would it be better to wait? No. I doubt there's going to be an Omicron-specific boost. We might have another variant by then. So the vaccine we have now is great for Omicron. Get it. That there, I'm 100% about that. Don't wait for a variant-specific boost because I don't know if you'll ever have one. When we get vaccinated, it's not just about ourselves, it's about the community at large. That's true. People you don't know, you're helping protect. I've always thought you have to be a bit altruistic. Unfortunately, not everyone is, right? I mean, when I used to drive a lot, I saw it every day. People cut you off left and right. They don't care. The other day, and I'll tell you what altruism is. The other day I was on the subway, and across from me is a young lady sitting, and next to her a drunk guy or drugged out guy was sleeping, and he was rolling all over the bench and getting close to pushing her off, and she was obviously upset, so I told her to take my seat across the way. That's the kind of thing that I do spontaneously because it helps. I can tell it helps someone, and it doesn't affect me in any way, but even if I gave up my seat, I don't care. That's altruism. I wish more people did that. It's not a big deal, but boy, it made me feel good, right? Because she was clearly relieved. Why does Amy think it'll take 5 years? I think mainly, well, you should ask her because I don't want to speak for her, but most of the world is going to take that long to get vaccinated, and that's what's going to stop this, vaccination. Don't doubt it, folks. Vaccination will end this pandemic. I don't know what's going on in China. I don't know about boosting. I don't know about variants. The last I heard, they have very low levels of infection. But if anyone's there, let us know. And the immunocompromised do matter, Tricia. I see your comment there. They do matter. They're people. One person matters. And so people are working to figure out what to do for them. There's a lot of immunocompromised people out there, actually. But still, they do matter. Was in the Moderna trial. Oh, I answered this, I think. Right. Will there be a second booster? There might be, but I would not condone it unless I see the data, just as I didn't accept the first one without the data. So why would you want? Why would you want to do that? So, boy, if they say another booster, I'll strenuously object, and we can talk about it here. Who are some other people who communicate to the public that we can trust? Frankly, you two and Twitter enough, but can you recommend anyone else? I'm going to let others do that. I think the Microbe TV crew, all the podcasters are really good at communicating to the public. I don't consume a lot of other communicators. And why, you might ask? Because I want to do my own thing. I want to develop my own style, develop my own solutions. I think this is the sort of thing that you do on your own, like teaching. I certainly was taught by a lot of people over the years, and I learned from their successes and failures, but I developed my own style, and now I don't watch other people teach. I'm sure I could improve, but I want to just do it myself. So you guys tell us who you think are good communicators. Netherlands went from 6 to 3 months because of Omicron. I just don't think that's useful. I think you should let the antibody reaction mature. And you're going to get yourself needing another boost, right? Is there data on the need to boost after getting COVID? No, but I think we have enough data now to say that if you are vaccinated and get COVID, you're fine. The infection has boosted you sufficiently, so you don't need to get boosted. And why is it too early to get it after 5 versus 6? Look, as I said, it's not a black and white issue. The switch doesn't go on at 6 months. It's gradual. If you want the maximum, in the study that was done, here's the best reason. The study looked at people 6 months, and they found their antibody response was high and broad, could neutralize a lot of variants. They didn't look at 5 months, so it's probably going to be lower. How much? We don't know. So you could probably be okay at 5 months. But I don't know about 4 and 3. It's getting lower and lower each time, because if you looked at 2 months, it was lower than at 6, substantially lower, higher than after the first 2 boosts only. So If you can't do otherwise, do 5. But if you can wait, do 6. I got the first Pfizer December 1st, got in an argument with the pharmacist about coming in 21 days. I said no, at least 3 months. He rolled his eyeballs and told me the official recommendation is 21 days. So this is the problem, Janine, that we are in. The vaccines have been licensed based on 3 to 4 week boosts, right? First and second. And you can't change that. So you have to do that and then get a third dose 6 months later, unfortunately. Until they change it, which I'm not sure they're going to do, right? Amy says 6 to 9 months. Does that mean affinity maturation occurs even as far out as 8? So that's a good question. Crotty has suggested that that maturation can continue beyond 6 months, depending on the antigen, right? The specific antigen, the timing, and so forth. So it's possible, yeah. Did I miss something in Canada? Other study about increase in disease death without the third? No, just antibodies. You didn't miss something. There's no disease studies that I would believe. You can find some out there, but the studies that convinced me were based only on antibodies. I would like to see disease, but that's going to be a while, so I thought it was worth to get the third dose now. If you were to take a wild guess, what would you guess the next? Oh, so the next is going to be an influenza virus for sure. We haven't had an influenza pandemic since 2009, and I think we're due. And it might be an H2N2. I could be wrong on that, but I'll bet it's an influenza virus. Okay, so the next question I'm preparing for Lucas, is there any TWIV with Professor Paul E. Turner? Yeah, it's a TWIVO. TWIVO number 44. Nels and I interviewed Paul at an ASM meeting. It's really good. You should go listen to it. I know vaccines are a clear benefit to high-risk people. How do you quantify the benefits for healthy younger people? So many younger people, certainly the death goes down as you get younger, but it's not zero ever, right? So death is a risk. And you can also get long COVID, even with a mild infection. So I would say long COVID, if I were a young person, long COVID would be it, because I know kids in high school who are track runners and they get COVID and then they don't recover and they can't run anymore. The vaccination will cut that rate down substantially. So I really think that's the reason. CBC News reported UK cases doubling 16 hours. I don't know if the number is right, but yeah, cases are doubling rapidly. Part of the problem is that there are a lot of unvaccinated people, and also that the vaccine doesn't prevent infection. And if you just randomly test people without symptoms, you're going to find some people are PCR positive. It doesn't mean anything. Like all these universities closing here in the U.S., it does not mean anything. They shouldn't be closing. They should revise your strategies. Well, look at that. We got the 500 likes. What a great bunch. Is there much interest or curiosity within your fields or with either of you in regards to COVID and captive wildlife? Well, we did have a lovely TWIV with a lady from the San Diego Zoo, veterinarian, quite high up. Maybe she was the chief veterinarian, I don't remember, a former student of Kathy Spindler's. And she talked about their experience with COVID and gorillas. So I think it's quite interesting. We've done a couple of zoo-based episodes. We've done one on TWIP also. We had the director of all the zoos in New York City, Paul Calley, on one TWIP. Very interested and would love to hear more. I'm also interested in non-captive wildlife, and I'm going to have the guy from Penn State who worked on the deer outbreak on TWIV next year. Is there a test that can distinguish between being infected versus vaccinated? So yes, if you look for antibodies against both spike and nucleocapsid at the same time, if you've been vaccinated with most of the vaccines, mRNA, vectored, you will only have antibodies against the spike. If you've been infected, you will have antibodies against spike and N protein. Thanks, Ian. Really appreciate it. As we all create variations if infected, how is a SNP, a variant, a strain determined? How is genome identified? If I isolate a virus from 100 people in a room, well, let's not say a room, from 100 different places in my state, they'll all be slightly different. They'll all be variants because they're not going to have the same genome sequence. So those are variants, and they contain one or multiple SNPs, single nucleotide polymorphisms. That's the right way to call it. A strain is something, a virus variant that has a very different biological property that has to be determined by laboratory work, not by field investigation, not by looking at epidemiological data. And it takes time to do that, and it hasn't been done yet for SARS-CoV-2. By far, none of these variants have been shown to have distinct biological properties in the laboratory. But for some viruses in the past, we've done that, so we call them strains. Can you explain what the scientific process is so that scientists can figure out which people are most likely to catch COVID? Is there any way to know if figuring this out is getting close yet? Well, of course, the obvious and simple answer is if you're immune, you're less likely to get sick, right? You can be infected, but you're less likely to get sick. So immunity is a big deal, and not everyone's immunity is created equal. As I said earlier, if you give 100 people the vaccine, they're not all going to respond the same way. Some won't respond at all. Some will have high antibody and T cell levels. Some will have low. Some will have intermediate. So the immune response is a big determinant. And so all the genes that go into the immune response play a role as well. And people differ in them. We have slight differences in all of our genes from me to you and everyone else. And they can regulate how we respond to infectious agents. And figuring out what they are can be done because now we can do genome sequencing on people and we can try and see what changes associate with, say, severe COVID. It's being done. What you do is you take people, say, with severe COVID, and with mild COVID, you can take a little bit of blood and do their genome sequence and compare and see what differences go with having severe COVID. As you can imagine, it's not easy because any two people's genomes differ a lot just because they're mutated. I shouldn't say mutated, just because the DNA replication process is error-prone. So it takes a lot of samples to figure out that one or two changes that are responsible for severe disease. And there have been a few hints so far, but the work is far from done. Yes, looking for nucleocapsid antibodies can tell you if you've been infected. But if it's a long time, they may be below the limit of detection. There are probably still some there, but your assay may not be sensitive enough, yeah. My daughter was double vax and got infected, so I don't think she needs a booster. I think the combination of vaccination and infection is good, in my opinion. Oh, the Y equals MX plus B is a great equation. I love it. When I first learned it, I got it. One of the few math things I got right away. A new paper on myocarditis published in Nature raises questions about risks in men for Moderna in particular. Given greater efficacy, thoughts on potential Moderna booster. I don't really think there's a significant difference in efficacy. That's antibody levels. I don't think that's the whole story. I think the Moderna and Pfizer are more or less in the population level equivalent. So if you're concerned about Moderna, take a Pfizer. However, as Daniel Griffin says, this myocarditis, vaccine-induced myocarditis, is treatable. And it can resolve in a couple of days. So it doesn't seem like you're concerned. But if you're concerned, go ahead with the Pfizer. If Amy thought Moderna was good, she would have done. She got 2 Pfizers initially, and she got a 3rd Pfizer, and I think that says it all. I think you're discounting a large increase in infections in the general population will mathematically result in more hospitalizations and more deaths. It may not change percent, but absolute numbers. Yeah, well, that's true. That's absolutely true. But again, most of those are going to be in unvaccinated people. You know, really, if 90% of the world were vaccinated, we'd have none of this discussion. None of you would be here, which would make me sad, but happy that everyone is healthy. And really, that's all it takes, is to vaccinate most people. Would you know what is the proportion of multi-particle viruses relative to non-covirus? Multi-particle. I don't know what the proportion is, but many plant viruses, they package different parts of the genome in different particles. And that's all I can tell you. I don't know. In the overall scheme of things, I don't know what the proportion is. We're just talking about virus pastries now. Ontario classifies 6 months as 168 days. You know, we're not doing rocket science here. 6 months is 180 days, right? 168 is fine. As I said, it is not a switch, and that should be fine. Did Omicron knock us back a step or two? No, I don't think so. What's keeping us in the pandemic is failure to vaccinate in many places where people don't want to, and in many places where people don't have the vaccine. Is there a public health response anywhere that we have found impressive? Look, Guide Monkey, not everything is criticized, but you may find that because we talk about those the most, right? But yeah, we don't praise the good ones. And there have been many public health responses. For example, Wales has a wonderful testing program, the best in the world, that they implemented. I really like the early efforts by Trevor Bedford in Washington, where they took their flu surveillance data and started looking for SARS-CoV-2. I think China's response was draconian, but it certainly curtailed their outbreak. And there are others, but they don't last forever, right? Viruses can squeak through it. So I'm thinking of Singapore early on had a great response, but then they started having cases for other reasons. I want Amy's panettone. Well, I'm sure she'd be happy to send you the recipe. She gave me one yesterday. I have it upstairs. I haven't had it yet. But the word is that it's very good. Yes, don't you think Bat Zero is a great name for a pandemic movie? Yeah, go with it. Take it and run. But don't say it's your idea. The person who said it there, say it's their idea. The one thing that really bothers me, I love a good idea, but when someone else takes it, that really sucks. You know, just own up that someone else did it. A lot of the things you guys say, boy, I wish I said, but I'm not going to take them. So, Mark, you agree that a good scientist can make pastries. Yeah, that science is like cooking in a way. I was never big into cooking until early in this pandemic. I started cooking a lot, but then I've since gone back because this has really consumed me, all the things that I have been doing for communication. And I understand I should diversify, but I just like teaching so much I can't. Wasn't there a paper showed you can only get levels of ivermectin in vitro? So the levels you need in cells and culture are higher than you would get by the approved single dosing for parasites in humans. But you could do multiple dosing, and that's what a lot of people have done to raise the levels. Now, the problem is this is a drug that binds channels in the CNS. And so, as Amy said, there are going to be neurological side effects if you take too much. So you need to do a trial is underway. So, we're going to see at the doses that they're recommending whether it works or not. Can we justify young males voluntarily vaccinating except on altruistic duty basis? Or is myocarditis a greater risk for them? No, I think the myocarditis from COVID is a greater risk than from the vaccine. And if I were a young male, I would vaccinate simply to avoid long COVID and ruining your physical health for who knows how long, but maybe the rest of your life, right? Thank you, Kathy, for your contribution. I'm glad to make your life less stressful. Sometimes on these Q&A, we raise the stress level. Hopefully tonight it wasn't. But I do want to give you good information. And I tell you when I don't know something. I'm trying to be honest with you. I'm just trying to be a scientist. How does one variant compete another? By doing something better. You take two viruses and you throw them in a cell in a lab and one will grow better. It does something better. And you have to figure out what it's doing better. I can't assume what it is, but let's say, to make it simple, one virus makes faster RNA, so it can reproduce faster and it uses up all the raw materials in the cell. And so it will reach in a population a host before the other virus does, things like that. Will poly vaccines, that looks like a spell checker thingy. No, I think the current vaccines can take care of it. If we can get more people vaccinated, we will take care of the variant problem. The variants are an issue because so many people are infected. Still, that's it. Very simple. More hosts, more mutations. What we need is a truly incredible science communicator for the mainstream. Well, I'm not for the mainstream, right? But I want to build my company to be, and it may not be me, but somebody that I get in the future to work for me. What skills? They need to know science. I don't care how charming you are. If you don't know science, I don't give a crap what you do. And there are plenty of science communicators who are very charming, but you dig a little deep and they don't know squat. So I want a communicator that can do virology like I do, but be more charming so that we can attract more people. Thank you, Vincent, for your contribution. What kind of instant testing would you like to see? Instant. I think the idea of having a cheap paper strip that you put in your mouth and in 5 minutes tells you if you're antigen positive, that you could do every day for free, would be the ultimate. And everyone can have a pack of them at home. That would make a big deal. You don't have to go get PCRs done. And that was a promise last year, but it never came through. Do you know how far we are from having clinical tests to determine if a person needs a booster? Oh, no, that's actually something that Daniel Griffin asked for last week on his clinical update. He said we need to standardize antibody tests so that we can have quantitative tests that actually give you information about your immunity. And how long? I would say at least a year is going to be before we have those. What do you think about mandates? Lots of vaccines are mandated for school, for kids, right? And some people have problems with those. If the mandate is the reason many people aren't getting vaccinated because they want to make their own choice. So what would happen if you removed mandates? Would half of the people get vaccinated? So it depends on the answer to that. I just don't know if it's an impediment or not. When I was a kid, when I was 8, I went to Europe with my parents to visit Italy. We had to bring a vaccine passport for I think it was smallpox vaccine. That was the only one I remember. There might have been others. It was a yellow card that went inside your passport. If you didn't have it, you couldn't go. Nobody complained about it. It's if you wanted to travel, right? So now if you want to travel, you're going to have vaccine mandates. What about a workplace mandate? Well, some people don't want to be infected by you because you're not vaccinated. So it's a rat hole, frankly. Let me stick to the virology because I can't give you an answer. Unlike other VOCs, Omicron appears to have gained the ability to bind rodent ACE2. I think that's not correct. There are other VOCs that bind rodent ACE2. I thought that was a property of some other variants, so I have to check that. It's one of these changes. One of the previous variant changes was already identified by Ralph Barrick as allowing binding to rodent ACE2. It's not a big deal. I don't think he's right that it's the first, though, but I will check. Can we learn anything from statistics on Omicron? Well, you can learn something, but don't make generalizations to the world because they have a very different population from the rest of the world, right? It's a predominantly younger population, for one, and there are other differences. So wait till you get data from elsewhere. I think it's clear now that Omicron is fit. It's displacing Delta in many places. It's making inroads in the U.S. It's clear that it's very fit. So let's see what it's doing in other countries. Will there be return to normalcy in the spring? The Omicron doesn't change any of my estimates. What is the issue is how many people are vaccinated. And the U.S. is stuck, right, at around 70%, and that's the problem. And if we don't get above it, we're still going to have lots of cases until everyone gets infected or vaccinated. That's the bottom line. Thank you, Mark, for your contribution. Oh, don't worry about it. Every little bit helps. Appreciate it. Could the cultural practices, as we were discussing earlier, meat markets, live meat markets, be regulated? I think they could be, right? Maybe you don't have to eliminate them. I think that it's a big issue, right? If that's the source of these two outbreaks, SARS-1 and 2, that's a problem. But, people get very dug in to their practices, and I understand that. Should a teenager get a booster? Yes. It doesn't matter how old you are. If you had those first two closely spaced, it's not good. You need to get a third to correct for that. If SARS-CoV-2 is the outcome of a bunch of recombinations, how long do you think might have taken for it to be produced from the ancestral type? It's hard to know. It could have been produced sometime in 2019 and fortuitously encountered a human, right? Or it could have been made years earlier and then encountered a human. It took years to encounter a human. I have no idea. Could be either way. I've heard more about countries planning to give out third, even fourth doses in the last 2 weeks than I've heard on plans to provide one dose to lower income countries. Yes, the world is a mess. This is horrible. We should be helping other countries more. I know we're helping, but we don't need a fourth dose. That is rather selfish. Does the field of public health need a reformation in terms of methods? All right, so all I want to do is point you to Laurie Garrett, who, when she was on TWIV, said the problem with public health is that it's run by MDs who don't know public health. It needs to be run by epidemiologists who are specialists in public health. And you know who the head of the CDC is, right? I don't mean to criticize her, but she's not a public health person. She has other training as an MD in HIV, AIDS, and so forth. But I do think all the public health agencies, and she said, and Larry Garrett, that is, said in New York City, all the workers in the public health department used to be public health people. Now they're MDs pushing paper. So I think that's part of the problem. I believe her on that. I'm not an expert in it. What happens when you get the second dose too early? Well, what happens is you interrupted the maturation of the first, and now you give it a new boost, and you've got new cells, B cells, recognizing that antigen. You didn't allow the first to mature properly, so it starts over in a way. And a six-month dose corrects for that, because at 6 months, most of the maturation has occurred, or enough of it to give you good protection, as the data say. First and second doses of Moderna, 9 weeks. I think you should get a booster. You should get as close to 6 months as possible. Yeah. Is there any more info on the degree of illness caused by Omicron? No. Not at all. that everyone is saying, oh, it's milder. But look, 10 people said Delta was worse and 10 said it was not worse. So you can't believe anything about Omicron yet. Hey, look, I'd love it if it were milder, but I think it's too soon to tell. You'd think if I were a jerk, I'd say, hey, yeah, it is milder, but that would be so contradictory because I'm saying for Delta there wasn't enough data, and so now there isn't. So just wait. It's too soon. But that's what the media does. They take what the scientists say. The scientists think they have it right. They want to, a lot of them like to get their word out there. Unfortunately, they have a big ego, and it's not really useful at all. Like we said, I think last week, I think a lot of the sequence data shouldn't even be publicized. What can people do with it? They don't know what to do with it, as you see by many of the questions here tonight. We have 583 likes. That's pretty good. It's about half of all the people that are here. Thank you all for that. Really appreciate it. If human breast milk can protect babies, can a cow that recovered protect adults? That's a good question. There's certainly going to be antibodies in cow milk, but they're cow antibodies, and that can be a problem. So I'm not, if that worked, we would have done that. So I don't think that works. We used to take horse antibodies and give them to people as treatment, but now we have better ways to do that. The UK is in full panic mode over Omicron. Look, UK was in full panic mode over every variant that emerged. And in every case, the hospitalization and deaths were limited by the vaccine. So I think they overreact all the time. And I think it's suboptimal vaccination and clumps. Don't give me your overall vaccination rate. There are always clumps of people that are not. And that's where the virus is. Plus some event. Now we're going into the holidays. There's going to be a spike. Going back to school caused a spike. There's always a human reason for it. It's not to do with any particular variant. Rich Condit last week on Twiv, says, tell me if I'm right. Is this all really just humans behaving? It's not any particular variant? I said, yeah, that's what we've been saying. That's the op-ed Amy and I wrote for the Times a while ago. That's what we've been saying all along. Now, the problem is, Mario, if you listen to people talking on mass media. Why does mass media pick people? So they pick the CEO of Regeneron, they pick Neil deGrasse Tyson, they pick Peter Hotez, they pick Michael Osterholm, they pick cardiologists. These people have a big following, and they think they're going to get them more attention and that they know something. They think they know something. And they know something, but they don't know as much as a virologist. And how often do you hear Ralph Barrick on these programs? Very rarely. No, he's working in the lab. And they don't want to talk to him. He's quiet. He doesn't get excited. Same thing with us. It's a problem, and I'm trying to change it with Microbe TV. So help out. And you do want to help because you're here. Lot of booster questions tonight. It's great. It's a good thing that I settled my thoughts on boosters. If a person received J&J for the first and over 6 months later received an mRNA vaccine, should they get one more? So I think theoretically you're okay, but we don't have the actual data to show that. What we have are data that says if you get a booster at 6 months, it fixes having the first two doses too close together. So logically, yeah, if you got 2 doses 6 months apart, that should be fine. So in theory, you should be fine. But as I said to a few others, I'm a little hesitant to say go for it because we don't have those data. People with just 2 doses 6 months apart. I think you're probably okay, but I just want to be a little hesitant. I have a terrible premonition that corona is going to plague us for decades. The bats can't be stopped. No, I don't think that's right. Once a decade. How about that? We can make pan-corona antivirals and possibly vaccines, so why aren't we doing that? Could have done that after SARS-1. Okie dokie. Okay, more of that. I answered that one. What kind of antivirals could be stockpiled? Well, the Paxlovid and Molnupiravir are not pan-coronavirus. They do have some activity against others, so they could be modified to be more broadly acting, and you could come up with some new ones. But that would cost money, and probably the companies don't want to invest until that virus is upon us. That's how companies work. Did you read Michael Lynn, PhD, MD, thread on neutralizing antibodies boosters for J&J? Sounds like the best bet now is 2 mRNAs appropriately spaced after the first J&J. Well, that's his opinion. I think you could also do 2 appropriately spaced J&Js or an mRNA after the J&J. Unfortunately, we don't have the data to look at that, right? It's a real priority. That's a real problem. What's the proper spacing? 6 months between first and second dose. We should do a trial to make sure the antibodies you make neutralize broadly all the variants. That would be it, instead of wasting jab 3. But as far as I can tell from Daniel, they're not going to do that. All right, I should wrap this up. Let's speed through. I'm only halfway through the questions. I'm really sorry. But today's lessons, we screwed up the initial dosing. Now, there are people who say, Vinny, you're wrong. You actually need 3. But I'm not sure. I think you might need 2 if you space them apart. So get a 3rd dose if you were dosed in a compressed fashion, which most of you were. And the current wave is not due to Omicron, it's due to SARS-CoV-2. And ignore what the papers and the news outlets are saying. Just come here every Wednesday night and just go to TWIV. Listen to TWIV. We do 3 a week. You don't have to listen to them all. Just pick one. Listen to it. You'll learn. Westfield, thank you for your contribution. Do listen to the replay. What's the preferred vehicle? Well, here is convenient, but frankly, go over to microbe.tv/contribute, and you can use PayPal or Patreon. You can do a one time with PayPal. And now it's all tax deductible because we're a 501c3. I'm going to set up a web page for a better solution in the near future, very much like Parasites Without Borders, with a button, click donate. But in the meantime, microbe.tv/contribute. If Joe Rogan knew how funny Amy is, he'd love you. He'd love her. Yeah, I think he would, actually. She should go on her own, but she'd probably be a little nervous about that. Is there a TWIV where you talked about the counter-argument? Yes, there is. We went through it several times, and I don't have the episodes offhand. Maybe some of the moderators could get them for you. Otherwise, I would have to search them out for you. Thank you, Holm, for your contribution. Really appreciate it. Perturbator. That's cool. Yeah, if we have a fundraiser, we're going to have fundraisers for sure. We will have cookies. Yes, your lymph nodes are making mutant antibodies, but they're not really mutants. They're good, right? They're more high affinity ones. The lower affinity ones get tossed out. Yes, Cornell is going complete remote because every case is fully vaxxed. So they're just randomly testing kids and they're positive PCR. I bet most of them don't have any symptoms. I bet most of them are not even replicating virus. The virus just came in and hung around, and that's what they picked up by PCR. It's really a failure. But Cornell was a failure from the start in a way because the president didn't want to test early on, and we called her out on TWIV. Do you remember? And now they're failing by over-testing. Now, if Columbia shuts down, I'll be eating crow, I guess, but I have nothing to do with that. Thank you, Merle, for your contribution. I really appreciate it. There's a study last week we did on TWIV. Just go to microbe.tv/TWIV. All the bloody papers we talk about are there. You don't even have to listen. The references are there. It was Friday TWIV. We talked about a study out of Ontario that showed that the spacing was important. And today on TWIV, we did another study which looks at the similar thing. So check those out. Thanks, Tom. Thanks for your contribution. Thanks. I'm glad you liked the antiviral lecture. I've had many years to polish those up. And I really love doing them. Boy, do I like talking. Thanks, Sandy, for your contribution. Really appreciate it. I find this channel the most elucidating and most authoritative. Well, I'm glad it's elucidating. I'm not sure it's authoritative. People would claim that we're not, but we just do the science. We go by the data. So if you like it, come here. Most people don't. No problem about that. Kent, thank you for your contribution. Really appreciate it. Do I know Christian Drosten? Yes, he's been on TWIV twice. I think has good aspects, and I think he doesn't have the right interpretation of other aspects as well. So, but that's the case with most people, including me. You should ask Amy. My lecture on antivirals. I am teaching an online virology course, Virology Live, here on YouTube, and I did antivirals the other day. Check it out. It's all recorded. You know, you could listen to it. Just take some time from something else. Not meaning to be nasty or anything, but you could learn a lot from it. I think they're fun, frankly. If 8-year vaxxed in school anti-mask is 4-year unvaxxed safe from Omicron at home. A 4-year-old unvaxxed, well, is unsafe from SARS-CoV-2, right? Any SARS-CoV-2, not just Omicron. So it's a problem, yeah. This is a problem with not masking at school. They're not thinking this through clearly. I'm sorry, it's a real problem that you can't have your kids masking at home, right? I don't know what to tell you. Hopefully the kid is 5 very soon, right? And yes, I think many countries are going to have fully vaccinated at 3 doses now that we have more data. Let's see. I'm going to speed through these because I've kept the mods here a long time, and all of you, and I appreciate it. Right now, 900 people. Thank you, Ditching, for your contribution. Really appreciate it. Come back next week with your questions. I would love to stay longer, but I don't want to make you stay. I don't want to make the mods stay longer. It wouldn't be fair to them, but I could go another 2 hours for sure. Do you want the podcast transcribed and summarized? If you go to YouTube, they play a transcript alongside the video, and you could go to the end of the video so the whole transcript's there, and you could copy it and paste it into a document. Now, I guess I could do that too, but, seeing what I do, I don't know if I'd find time for it, and I'd fall behind very rapidly. Thank you, Don, for your contribution. It's very much appreciated that you have the faith in what we are doing to give us your hard-earned money. Thank you, Lisa, for your contribution. Really appreciate it. I'm glad we boosted your memory about the Fields Virology chapter. Yeah. So I'm revising the history of virology chapter for Fields Virology, the next edition, with Lynn Enquist. And I have been woefully behind. I'm trying to finish it this week, at least before the holidays. Oh, boy. Thank you for your contribution, James. Really appreciate it. Thank you, James, again, for your contribution. A different James, I'm sorry. Do I think there will be a variant that is completely vaccine resistant? No, because the T-cell epitopes, Amy, the parts of the virus that are recognized by T-cells in you, do not change. So the T-cells will always rescue you. Even though you might get an infection and mild symptoms or even moderate symptoms, you will still have T-cells, so there will be no variant that escapes the vaccine completely. Thank you, Nathan, for your contribution. Glad you like it. Hey, hello to McGill. Shout out. Amy was at McGill for a number of years. Thank you, Sam, for your contribution. Really appreciate it. I would very much like to have Trevor Bedford on. Yeah, I just have to get around to inviting him. I wish I had a producer who could do all that for me. Hopefully we raise money and I can hire at least a part-time producer to do that. Boy, a lot of questions here. I'm so sorry I can't get to them. Thank you, H. Zoo. My family in Wuhan are all boosted with Chinese vaccine. Contact tracing, health code is still in effect. A single positive case triggers an alert. Okay, thanks for the update. Appreciate it. So third doses are being given in China. Thank you, Overjoyed, for your contribution. I really appreciate it. Thank you, Elise. Love it. Thanks for your support of our work here and elsewhere. Thank you, Ron. I had J&J in April, called COVID in October. Should I get the booster? No, you're good. I think combination of infection and a dose is good. I always feel very badly at the end where I can't answer everyone's question. Thank you, Andy, for your contribution. What if I had COVID in March? Fully vaxxed. Is a third dose needed? No, you don't need a third dose. You guys are smart. You put your questions in a contribution because you know I have to speed through the others. Why not use mRNA to vaccinate against all coronas? Well, the common cold coronas are not much of a problem. By all coronas, do you mean all the ones in bats? Well, we don't even know how many there are there, so you'd have to find something in common to do that. Someone needs to go on to Joe Rogan. Well, as I said, he doesn't want us because we speak the truth. He had his chance. I was there in Austin. He was on the phone with Lex Friedman. He knew who I was. The name was mentioned. He didn't pick it up. So he doesn't want it because it's not going to give him what he wants. All right, folks, thanks so much for coming tonight. What a blast answering your questions. Thanks to the moderators tonight. Ozzy Cam, Les, Frank, Vanity Nutrition, Steph. Thank you all for doing a great job. Thank you, Violet, for your contribution. And please come back next week. I will be here with Amy on the 22nd, next Wednesday, for more Q&A. And come earlier so you can get your questions in. Really appreciate it. Thanks, everybody. Be safe and get vaccinated. Bye-bye.